Thank you, Tom, for this very kind introduction. I seem to have reached this stage in my career where there's danger that the introduction is more impressive than the talk itself, but let's see how that goes. I would also like to thank Alvin for giving me this opportunity to speak today. I only had to think about this opportunity for about a second. It's a great pleasure for me to be able to help and to make this day a little bit special under the circumstances. I would really like to help the place that carries our founder's name, the Max Perutz Labs. I would also like to help Tom. Tom was, I think, my second PhD student, and I've just learned that he's become deputy head over there, so I'm very proud of that. I'm now going to switch to the story and share my slides. Let's hope that that works like that. So we are celebrating Max Perutz, and here he is in his office. And I wanted to start with a little story that I remember very well. I was a young postdoc in the 1990s, end of 1990s. I had just arrived at the LMB, and I was attending a seminar. I can't quite remember what the seminar was about, but I can remember one particular incident. After the lecture, a voice emanated from the floor just in front of the speaker, asking an overly simplistic question. And it emerged that that voice was Max Perutz's voice. He was laying flat on the floor, trying to deal with a bad back, which he frequently suffered from. And I also remember that the speaker had great difficulty answering that simplistic question, because it turned out it was a very clever question that exposed the weakness in the reasoning. So I've learned from that two things: that being a bit eccentric is not a detriment to doing good science, and it may even be a help to think outside the box and doing things a bit differently. And I think Max was a great example of that. And I learned that often it's the simple questions that are the hardest to answer. And we all should try once in a while to go back to the really simple questions, because those are the ones that give important answers. So here's Max in his office, probably in the late 90s, I would say, maybe early 2000s. And he would have been 106 today. I've listed here the main contributions that I think he has made. He solved with John Kendrew the first two protein structures ever. He founded the LMB. He was a great science communicator, and I think it's really fair to say that he's one of the most overlooked giants of 20th century science, in fact. And you will see over the next few slides why I think it's a fair statement. It's also important to say that in his later life, he was not completely overlooked. If you look at the number of letters behind his name, that's a sort of UK specialty. He was very highly decorated, and he was a member of the Order of Merit. That's the highest possible honor the Queen can give to anyone, and there's only 24 people here in the UK who can carry that award at any point in time. So Max grew up in Vienna, as you all know, and he got a chemistry degree from Vienna in 1936. I think this picture here on the left is a great example how he was probably already quite interested in details in an early life. Here on the right, you can see that this is one of Max's own pictures, actually. He developed a fondness for mountaineering and for photography very early in life, and he continued that all his life. For reasons that are sort of fairly clear, he then decided to move to Cambridge, because he had learned that there's some important progress made by certain people here in Cambridge concerning biochemistry. And he ended up to work for his PhD with Bernal and the younger Bragg, Lawrence Bragg, on the structure of hemoglobin. And I think it is important to point out here that it was really Bernal who had the vision of that being important later, because it wasn't really clear how one would do this. And Lawrence Bragg really provided a lot of support over many, many years, and was very important for the story. I should also add, and I haven't put this on this slide, Bernal was not known for getting things done. So having Max Perutz there and one of the Bragg family, that was very important. Now then disaster struck. Hitler took over Austria in 1938. Max's parents had to flee to Switzerland and lost all their money on the way. And that meant that Max had lost financial support for his studies in Cambridge. So what he needed to do is he worked then on a project to earn some money, and that was on the conversion of snow into ice, combining his fondness for mountaineering with science. And this project actually meant that he, for all of his life, he kept quite interested in ice crystal formation and the science around that. Once World War II had started on Churchill's order, a lot of Austrians and Germans in Britain were interned and not to be able to interfere with the war effort. And that meant that Max was sent to Newfoundland. Luckily, he could return after a few months, and then was put to work on a project called Habakkuk, which is sort of a crazy idea where they wanted to make giant aircraft carriers in the middle of the Atlantic made from ice and wood chips. And it seems like a crazy idea. It was never put into action, but the idea was, I think, that Max's expert knowledge on ice would somehow help with this. Somehow this was all done under a meat market in London in some secret location. After the war ended, the MRC recognized, and this is the UK's Medical Research Council, recognized the importance of some of these attempts and founded the unit for the study of the molecular structure of biological systems. It's sort of the beginning of the idea of molecular biology. And importantly, you can see here, Max recruited some very important people. Francis Crick was actually his PhD student, James Watson from America, and then John Kendrew and Hugh Huxley. And all of this, it's important for me to say this, was still happening in the physics department of Cambridge University, which then was the old Cavendish laboratory, which you see a picture of here. And then what followed was sort of the annus mirabilis, 1953, where three of the projects that had been started after the war really came to fruition. One was that Max and the people around him found a way to convert these diffraction images from the crystals into actual electron density, although only at low resolution. But the principle had been set, and it was now possible to determine structure. It was very clear. This is really Max's greatest scientific achievement. And then the same year, as you know, Watson and Crick discovered the structure of DNA, and also Hugh Huxley published his filament model of muscle contraction. And it's very clear that this new unit was extremely agile and extremely successful.
The diffraction pattern shows only the intensity of the waves. To work out the molecular structure, it is also necessary to know the phase.
Bragg and I had a lot of discussions about other ways of solving the phase problem until I hit upon the right one in 1953, so 16 years after I started. And that was the heavy atom method. Now, in 1939, Bernal suggested that this might also be applied to proteins. And we all thought that adding one heavy atom or two heavy atoms to a molecule with 10,000 light atoms would make no measurable difference to the intensities of the diffraction pattern. So I did these measurements, and to my surprise, I found that less than 1% of the atoms in the molecule contributed, and that if I added a heavy atom, all its electrons would be concentrated at a point. They would all scatter in phase, and so they should make a measurable difference. It was really one of the most moving moments in my whole scientific career.
In 1957, the unit was then renamed into the Unit for Molecular Biology. And because the physicists were still not entirely convinced that what Max's little unit was doing was important, they had to move out into this hut here. And that turned out to be a very major decision that went very wrong, because it separated the unit from the university, and that has led historically to a lot of problems between the two organizations. The same year, Sidney Brenner was recruited, and Francis Crick published his central dogma, which meant that information flow in biology will go principally from DNA to RNA to protein and not back. And then in 1962, the MRC funded an entirely new building for the new MRC Laboratory of Molecular Biology, which became an MRC Institute, and Aaron Klug was funded. Tom will still remember working there. He used to work in one of those rooms down there. And that is the famous picture of the leadership team of the new MRC LMB, because in that year, Fred Sanger had been recruited from the university, and famously he then later developed DNA sequencing. All those people have won Nobel Prizes except Hugh Huxley. But then, while all this was sort of going very well and this was extremely successful, Max's own project was not actually going so well. John Kendrew, with his help, had solved the structure of myoglobin at 6 angstrom in 1957, and then at 2 angstroms a couple of years later. And that meant that Max had really lost out on solving the first protein structure. And you can see here in this picture in the bottom right that, you know, the resolution of the model of hemoglobin and of myoglobin are distinctly different resolutions, and John's face here shows that very clearly. But then together they did win the Nobel Prize in 1962. This was for Perutz, Kendrew, and Watson and Crick. And in 1970, finally, the structure of hemoglobin and atomic resolution 2.8 angstroms was determined. It was the second protein structure ever to have been finished. And here's Max proudly fiddling with his model. So that's sort of Max's science, but most people will probably know also that Max was a very gifted science communicator. And I think for all of us, it's very important to remember that as scientists, we have a duty to communicate well what we're doing and why we're doing it. And Max was particularly effective. He spent many years reviewing for the New York Review of Books. That sounds sort of slightly obscure, but actually it's a very important publication that deals with many different aspects, not just with books. And here on the right, you can see that Max was often on television trying to explain how protein folding works and the structure of DNA and all sorts of findings that his unit or his institute was involved with. If you watch these episodes now, it's amazing how much detail television programs contained in those days compared to today. Sadly, Max died on the 6th of February 2002, aged 87. Just to summarize his scientific achievements, he developed the method Isomorphous replacement to solve protein structures by crystallography. He helped to solve the first and solve the second protein structure ever. He, together with John Kendrew, showed that there's something like structural homology or similarity, because myoglobin and hemoglobin turn out to have exactly the same fold. Max spent many years after his first structure to deduce allosteric regulation, as you can see there on the right in this little morph, that upon oxygen binding, these proteins change their shape and change oxygen affinity in the other half of the molecule. And that's something that nowadays is extremely important in many, many systems. And also, hemoglobin really showed the power of structure for what we would now call precision medicine very, very early on, because sickle cell anemia is caused by point mutation in hemoglobin, as was shown mostly by Ingram in 1956. And the structure immediately showed how that would actually lead to filament formation, which is the cause of the disease. So I think it's sort of absolutely amazing string of achievements that make him one of the really great people of science of all time. But his legacy sort of goes further in the sense that with recruiting these amazing people, founding an important institute, and thinking about it very clearly, he laid the foundations of structural and indeed molecular biology to a large extent. His science communication, as I said, was exemplary, and he showed, I think, to most people that we need to trust to apply the rules of chemistry and physics if we want to understand biology most of the time. And he founded LMB. And he has sort of shown the number of structures that are being solved worldwide. So the light blue is the cumulative number of structures solved. Max started here somewhere far on the left. I started in about 1992, but you can see we have now almost 200,000 structures, and there's no end in sight. It's an amazing technology, and without it, we wouldn't understand half of what we understand now. I've mentioned the LMB a few times now. That's what it looks like today. We moved into a new building in 2013. It's much bigger now, but we really try to maintain some of the features of the old lab that Max really was very keen on. For example, with his wife Gisela, he started this idea of having lunch at lunchtime. It's something very un-British to do, but he, as a continental, really wanted that, and he started this idea that people come together once a day, sit together and have a chat, and with a new building, we really try to still do that. And there's many other features, such as open door policy, that Max was very, very keen on. All of that is still alive today. As Tom already mentioned, the LMB has managed to win 12 Nobel Prizes so far. And I don't want to sort of brag here. What I wanted to point out is that this prize here in 2017, jointly given to Richard Henderson, is very important. And I will sort of use the rest of my talk when I talk a bit about science, that you sort of realize how, since Max's day, structural biology really has moved on a big way in terms of method, mostly caused by the arrival of cryo-electron microscopy, but that the basic principles that Max really believed in, the use of chemistry and physics to understand biology at a mechanistic level, that really has not changed at all. This is the prize, and I made that prediction already a number of years ago. Electron microscopy will really be the dominant method in structural biology, and you will see why that is over the next few slides. So I'm going to talk you through 3 projects that come from my group that I've picked to highlight how structural biology has moved on, partly because of EM, but also partly because we understand a bit more. And the first project that I wanted to show is a large protein called Thyroglobulin that has never been crystallized. So it has been known for many, many years, but only with cryo-EM could that be determined. Thyroglobulin is a large protein of 600 something kilodaltons as a dimer. It's heavily glycosylated, and it resides in the thyroid gland in all vertebrates, where it accumulates to very high concentrations of about 100 milligrams per milliliter. It's been known for many, many years, and it's used as a molecular weight standard because of its abundance in many labs. Thyroglobulin is needed to make thyroid hormones. I don't want to give you too many details, but these are the 2 hormones that the gland produces and that control expression of many genes in all the cells in your body. So pretty much every cell in your body will have a receptor for these hormones in the nucleus that then will switch certain genes on and off. And all growth, most metabolism, brain development, and heart rate are very clearly controlled by these hormones. And when that goes wrong, then you're really not in a good state. And it turns out that 5% of the world's population suffer from some form of thyroid disease or anomaly. These hormones are made in a very peculiar way. This is something that we haven't found out. This is something that's sort of the foundation of sort of modern medicine, where this was figured out. These follicles here in the thyroid gland, they have an extracellular volume on the inside, surrounded by a sphere of cells in a single layer. These secrete thyroglobulin protein into the follicle, and then some of the tyrosine side chains on the protein react with iodide, hydrogen peroxide, to form these iodinated tyrosines. And then you get a radical reaction in which one of the rings of the tyrosine side chains jumps onto the other, and you get the hormone here on the side. This is a chemical reaction that cannot happen in cells because it requires radical chemistry. After this has happened, the protein is re-imported into the cells, cleaved, and then the hormone is released, which is basically made out of two side chains. This reaction here is the reason why all human beings need iodine. And as you know, in some environments, there isn't enough of it, and then you suffer from a deficiency in hormones. So this is a peculiar protein substrate. It's not an enzyme. It actually is used to make something as a substrate. When we started, it was known that there's 4 acceptor tyrosines only. This was done by mass spec, but the nature of the donors had not been determined, and there was no structure available. So we wanted to find out. So we had a look at the protein. And first, we needed to get the protein. You can purify this. You can get what's called goiter tissue. So these are from patients that have largely expanded glands because of a deficiency or because of something not being quite right. That protein is very heterogeneous. It's poorly iodinated and partially degraded and contains some hormones. It's not ideal for starting some studies. We then developed an expression system in mammalian HEK cells that gives very beautiful protein that is clearly not iodinated. The cryo-EM progressed really nicely. You can see here the endogenous protein is very similar to the recombinant protein, showing us that the recombinant expression system is really capable of producing the correct material. The structure determination went through a lot of cycles, as it always does with cryo-EM. For those who know, this was all done on graphene oxide that makes the protein absorb a bit more and increases the concentration. And we needed a lot of tricks to get the resolution up in the outer bits here, but that's not really important for the biological story. The protein is large, so it took us about a year to build the atomic model. This is all de novo. There was no previous structure available, and you can see here there's a number of thyroglobulin-like repeats that follow particular folds. None of this is really interesting for the story. Just to say that we built about 2,500 residues, which is about 93% of the sequence. And here's an overview of the structure and the density. So you can clearly see the dimer here in gray and in blue, chain A and chain B. And this is quite obvious immediately. The dimer is not very clearly separated. So one of the chains intertwines very heavily with the other. That made model building very, very difficult. Here you see the glycans. Some of them are actually structural, like this one, for example, where the sugars are really part of the protein fold. So if you were to not glycosylate this protein, it wouldn't fold correctly. And you can see here the intertwined nature. So if you start from the N-terminal domain and then go through the core, the flap around here, the arm, to the C-terminal domain, then you can see how all this is sort of pretty messed up. So let's just cut the chase and find out about how the hormones are actually formed. So I said 4 of the acceptors had already been identified by mass spec. So how do we find the donors? Now that we have a structure and we know that for hormones to form, 2 tyrosine side chains have to come together, what we can do is we can look in the structure and have a look where these things are. So let's have a look at side A here. So you can clearly see where the acceptor is. It is here. Then there's a donor very close. That's very, very good. But then there's also one here on this side. So there may well be 2. If you just look in side B, there's the acceptor that was known. And then there's only one donor right next to that. And then there's side D, sorry. There's side D down here, again, where this is very clear, there's an acceptor and there's only one donor next to them. Side C is is disordered and we don't see it. So that's just looking at what tyrosine pairs could be involved in hormone formation, just looking at the structure. In order to be sure about that, we needed to really investigate this mutagenesis. So what we did is we used our expression system in order to look at mutants, and then looked at the production of hormones. For that, we adopted an ELISA assay that just competes off HRP fused T4. And when we do that and add iodide and the oxidase to make hydrogen peroxide, then we get very good hormone formation. If you don't add iodide or don't add oxidase, you get nothing. This here is the endogenous material without anything added. You can see it already comes with some hormones, and all the controls are negative. So we can clearly measure the production of hormone in vitro from the protein by just adding iodide and oxidase. And now we can mutate all the different acceptors and all the different donors and figure out under what circumstances do we get to hormone formation. Here's the wild type signal. And if we mutate all 4 acceptors away, we get no hormone formation. If we only mutate 4 donors, remember site A had 2 alternative ones, so we only mutate 1 of them, we still get some hormone formation. And this is for the other 4. But if we mutate away all 5, those 2 and all the other ones, we again get none. Meaning that we now have clearly figured out there's 4 acceptors and 5 donor tyrosines in 4 hormonogenic sites per monomer. It is also important to test whether all the sites are active. We can do this by actually putting back each individual site, and you can see, although they're not exactly equal, they all make about a quarter of the hormone per monomer. So we have figured out from this that really it is the proximity of the tyrosines that's important for hormone formation. The residues have to be exposed. You can see in the little movie there that they all are on the surface of this large protein, and they all are in flexible regions for efficient coupling probably. This was quite difficult during structure determination because these are all loops that are not very well ordered actually. So it turns out there's not really a structured active site. It's really just a substrate for an efficient chemical reaction to occur. In order to really drive home our understanding of this, we wanted to go further. So we asked, can we make hormones from another protein by adding tyrosine residues in exactly the same way as we found it on thyroglobulin? For that, we chose bacterial MBP, completely unrelated protein. We chose that for 3 reasons. One is there's already a tyrosine pair in one of the loops, which is quite flexible. And the idea was this might already make hormone just like that. The second idea was that we could maybe take this tyrosine here and then put the tyrosine in this position, and maybe they make hormones, although not really thinking they would be able to do this because there's no flexibility because they're both in secondary structure. The third idea was to add tyrosines to the C terminus, which is here, and then be able to couple that with that tyrosine. And you can see that all on this slide here. Sorry, this is a bit complicated. So we had 2 different concentrations of protein used, 1-fold and 10-fold more than thyroglobulin to get more signal. If you use the wild type protein, you get no signal compared to thyroglobulin, but in a 10 times higher concentration, we do get some signal. This goes away if you mutate one of these residues here. So clearly showing that the residual hormone formation comes from this site here. If we, in addition to that, add a tyrosine on this arginine, then we don't get any extra production to the wild type, meaning this site doesn't really produce anything more. If we add a short peptide with the tyrosine here to the C terminus, again, we don't get anything more. But if we add a much longer version, which you can see here in detail, then we get as much hormone formation from 1 molecule of MBP, from 1 molecule of MBP compared to 1 site in thyroglobulin. Really driving home the message that this is really not very enzymatic or specific. This is really just 2 tyrosines that are in nearly antiparallel arrangement, close together in a fairly flexible region. That's all you need in order to make T4 thyroid hormone. And this is all summarized here. So it really is a giant disposable substrate for a pretty dramatic radical chemical reaction. You will probably ask me at the end, so why do you need, 2,500 residues to make 7 little hormones? The reason is that because iodine is so limited in the environment, our bodies need to be able to store it. So what this protein actually does, in addition to the hormone formation, is that all the other tyrosines get iodinated just in case that no iodine is available for the next year or so. So that's one of the reasons. And the other reason is that this protein lives in an extracellular environment that's very harsh, and the protein needs to be very, very stable. Okay, so that was the thyroglobulin story. That's showing how with cryo-EM we now have access to structures that really we didn't have access to before at all. I then switched to our work on archaeal and bacterial S layers. That's really about how we can use electrons to probe proteins much more in their natural environment, in cells, or in this case on cells. What are S layers, for those who don't know? So most archaea and some bacteria have on the very outside a layer of essentially 2D crystallized protein. This was discovered in 1953, and it's a very, very common feature on many microorganisms. There's a great variety. They can be inserted into into one of the membranes, or just the outer leaflet, or they can be bound to the cell wall, and there's many, many variations of that. And S layers, as I told you, are 2D crystallized, and there's all sorts of different symmetries available. Most common is hexagonal symmetry, but there's all sorts of other versions as well. So it's it can a little bit be compared to coat proteins of viruses. They have very simple principles, but so far there doesn't seem to be any connection through evolution between them. So despite many attempts over many decades, in fact, by many groups, no atomic understanding of any S layer lattice had been obtained, and we wanted to change that. So we looked at a particular freshwater bacterium, Colobacter crucentus. Why did we do that? Well, it has an S layer. So you can see here the envelope through a tomogram. We start from the inside, so there's the inside of the cell here, then there's the inner membrane, then there's the cell wall, the peptidoglycan layer, then there's the outer membrane, and then outside that, you have the S layer. And we wanted to obtain a structure of this layer here to see how the cell really protects itself from the environment by using this two-dimensional protein sheet. Colobacter is quite special. It's a model organism that's used to investigate development in bacteria, because what these bacteria do is they make two different cell types. They make a stalked cell with these little holdfast at the end that attaches the surfaces. They make swarmer cells that have a flagellum for swimming. And what this enables these organisms is hedging the environment. So maybe in a pond, it's better to sit and wait for something to come along. These cells, the stalked cells, do that. Or maybe it's better to swim around to find some food, and the swarmer cells do that. That's not interesting here. What's interesting is that it's these stalks here that are a bit thinner than the cells, and we can use that for imaging. Just before I show you that, just a picture of an average of this envelope. You see a little bit more. So this is the cell, the inside of the cell here. The inner membrane is down here. Cell wall, can't really see that here. Then there's the outer membrane, and that's the S layer. You can already see that the S layer has an outer coat and some inner domains here that will become a bit important later. Here's the stalk, and here's the reason why we chose that. It's a beautiful picture. There's the molecular glue here that attaches the cells to surfaces, and the stalk is much thinner. So it's about 100 nanometers, that sort of thing, and that makes it possible to image with electrons at much higher resolution, and you would be able to image a whole cell. You can see it gets a bit black here. The inside of the cell, it would be very difficult to get resolution imaging. And in order to push this a bit further, we really needed to use a mutant that removes these stalks, so we can image them very clearly. That exists, and then it leads to tomograms like this. So this is a three-dimensional volume of one of these stalks, and we can then use a computer program in order to find the positions of all the hexamers on these tubes and average that together. And here you can then get the shape of one of these S-layer proteins. So it's basically a method called cryo-electron tomography with sub-thermogram averaging. So what we do is we first image the stalk in 3 dimensions to get a volume, then pick little sub-volumes out of that big volume and average these volumes together in 3 dimensions rather than 2 dimensions. It's a very important difference to single particle electron microscopy. Having done that, and we've had tens of thousands of these little sub-volumes, we could then get a map at about 7.4 angstroms resolution. Tanmay has, with his own group now, has pushed this much further, but this is sort of enough for what I want to show you here. You can clearly see this this hexameric structure, and you start to see even secondary structure here in the section to the map, which we now know correspond to beta sheets actually. So it's sort of difficult to a particularly difficult structure to visualize because beta sheets are the most difficult to resolve. This is the side view. So if you remember, that's the outer layer of the cells. The outside is here, the inside is here. And these are these inner domains that somehow attach probably the layer to the cell. So that's where we got stuck. So how do you continue? What can you do now in order to get to atomic understanding? For that, we really had to go back to Max's old technology. Crystallography came to the rescue. So don't forget crystallography. It can still be very important and very powerful. So we looked at that protein in vitro, and I got really worried because this is a really strange protein. If you look at it, it has almost 19% threonines. That's very, very unusual. And it has, if you look at the self matrix, it has a lot of internal repeats that worry people who want to crystallize. It might indicate disorder or things that are difficult to crystallize. However, we could crystallize this protein, and it turns out that when we dissolve the crystals, there actually there was a proteolysis step needed, which we then could reclone, and this was sort of a serendipitous thing to happen. And here's the structure. You can clearly see it's a beta helical protein. It's about 30 turns, right-handed, and the gray dots here are calcium ions. So that's something that was known, that these proteins strictly require calcium both for their folding and for the formation of the S layer. So now we know where they are. How do we know they're calcium ions? We actually used a very modern method, which is long wavelength anomalous dispersion at a special beamline that operates in vacuum at a diamond. And doing that, we can directly determine what elements we're looking at. And you can see here that all the yellow balls are demonstrated to be calcium. So we're very sure about that. Why do we need these calciums? Turns out there's bits of RTX fold in here. So you can see these are special loops that are very, very negatively charged, sorry. And then you have these calcium ions here that stabilize that. And if you look at the structure where these calcium ions are, they tend to be in loop regions, making this protein probably very, very, very stable, much more stable than you could do without these ions. It turns out that we can boil this protein in 100 degrees and it does not unfold. Why is there all these threonines in the structure? And you can see here the top view of the beta helix, and you might be able to see this here. There's these rows of threonines on the surface. For those who know protein structure a little bit, this is actually something that has been seen before in antifreeze proteins. So these are proteins that stop water from freezing. And it's very tempting to speculate that this is exactly the same mechanism here, that a freshwater bacterium might want to stop water around it from freezing because it would kill it. So this is something we are testing right now, and it's a very promising hypothesis. It might just be about salvation, that you need these hydrogen networks with the surroundings for the cells to become highly soluble, but it's tempting to postulate that it's maybe more than that. It may just be about freezing as well. So we were quite lucky. The crystals also contained the hexamer. So this is just the full asymmetric unit here, and I don't need to tell you that looks very similar to what we saw by EM. So what we can now do is we can take the hexamer we have from crystallography at atomic resolution and can put this into the 7 angstrom map we got from cryo-electron microscopy from the stalks that come directly from cells. And you can see here that fits really, really beautifully. And even some of the smaller details come out of the EM density very, very clearly. So we are very sure that the structure that was solved as a hexamer by crystallography is the same as it is on the cells. It's also clear that when we crystallized the protein, that this domain was proteolyzed off, and that's the reason why it's actually crystallized in the end. So I showed you this picture before. So we now know the structure of this layer here, and the structure of this N-terminal domain has just been solved by Tanmay, and they show that this is actually the LPS binding domain. So LPS is the lipid in the outer leaflet of the outer membrane that links the S layer to the cell. So we are now we have a good understanding now how this S layer is actually attached to the cell surface. So how about the lattice itself? Again, we were very lucky that it turns out that the crystals contained a complete layer of this. So actually the crystals contained this surface as we have it on cells. You can see this here. So the packing in the crystals is identical compared to the packing as we have it in the sub-tomogram average. So the S layer was solved essentially in situ to atomic resolution by combining the two methods, by combining the crystallography and the in situ sub-tomogram averaging. So because time is moving fast, I'm just going to switch to the last story, and this is meant to highlight that sort of what I said earlier about cryo-EM becoming extremely powerful, and it will become very powerful because in addition to giving us access to lots of structures and big structures and heterogeneous sample and so on, it also gives us some access to dynamics. Although this example I'm showing is not exactly showing that, but through very large data sets and sorting out different subsets of particles, it will be possible to get access to quite complicated dynamic processes that crystallography has struggled with quite a lot. So this protein is a DNA motor. It's called FTSK. It's again bacterial. And this is sort of a little bit of bacterial cell biology that I need to explain to you, why this protein is a DNA motor. Sometimes in E. coli, during replication, an accident happens and a recombination fuses the two circular chromosomes that should go into the two daughter cells into one big circle, which is shown here. It's called a dimeric chromosome. So instead of having two circles, you have one really big one. That's deadly, and both of these cells would die. If there wasn't a repair mechanism to deal with this, the repair mechanism goes like that. So you have these COPS sites here. These are DNA sequences that tell the cell which way you need to go in order to align these diff sites here. These are two different DNA sequences until they are aligned in the middle. And these arrows here are read by the pump itself, by FTSK, that then pumps the DNA until the two diff sites are in the middle. And that's shown here. So once it's happening, a recombinase is recruited, and the two chromosomes or nucleoids are disentangled. So that's what this pump is for. You don't need to remember any of this. It turns out that this is a very, very fast and very efficient and completely sequence unspecific double-stranded DNA motor. It's part of the divisome, so it's coupled to cell division. That's not so important here. What is important is that the N-terminal domain is involved in cell division. It's the C-terminal domain that does the pumping, and that's what we are actually looking at here. It's a fairly large protein, so the N-terminus, it's transmembrane and anchors it to the divisome. It's the alpha, beta, gamma domains that do the translocation. And there's a little sort of interesting detail here that has been worked out a few years ago. The gamma domain reads the COPS sequences. So the COPS sequences tell the protein which way to go by binding the gamma domains, and then the motor can only be loaded in one direction based on the length of the linkers here. You would not be able to load this on the other side. So basically, it's the loader that loads the motor, which is the hexameric ring, onto the DNA, only in one particular orientation. And what then happens is that the translocase starts, the DNA is sucked in this way, and these things are stripped off. The gamma domains are stripped off. So what I want to show you now is that I think we have determined what the molecular mechanism is of the translocation reaction itself, not the loading, not anything about cell division, just about the DNA translocation. So first, in order to actually look at the sample while it is translocating DNA, we need to do something because it's very, very fast. You can see here, it's the fastest, one of the fastest ATPases known, and certainly the fastest DNA motor on RNA motor. It hydrolyzes about 2,500 ATPs per second. So if you do a quick calculation, if you put one millimolar into a test tube with a reasonable protein concentration, there's no way that you could ever get a sample that isn't completely ADP. And this is exactly what we see here. So we look with NMR at the phosphor signal of the phosphates and the nucleotides directly. There's no labeling. You can see the ATP sample is purely ADP, no ATP. But if we use ATP gamma S, which is slowly hydrolyzable, you can see that over a reasonable time frame, we get hydrolysis. So this shows two things. The protein is active with ATP gamma S, and it is doing this in a time scale that is accessible for cryo preparation. So we also looked at other samples with mononucleotide, with ADP and AMP-PNP. These are not so interesting because these are obviously not physiological, and they're also completely symmetrical, which cannot be right. If you look at the ATP gamma S sample, now this is very asymmetric, and there's many reasons that I'm not explaining here that make us believe that this is really the translocating state. So we looked at the hexameric motor loaded onto DNA while hydrolyzing ATP gamma S. This is the EM map we get out of it. I just wanted to show this here a little bit, how good these maps can be. And this is although it's only 3.6, it's extremely easy to interpret. So the important thing here, as argument why this is a translocating state, is that in the one structure we determined, we actually see all three different nucleotide states. So we see some subunits in triphosphate, in ATP, gamma S. We see two in ADP, which presumably have been hydrolyzed because that hasn't been added. And we see some subunit that has clearly is clearly in exchange, so that the gamma phosphate has only half occupancy. So that means that we can from this deduce that the protein, as we look at it, is actually working, and we can deduce the direction because this is going to be the next nucleotide that will be hydrolyzed, and this one is the next one that will be exchanged. So it goes this way around, clockwise when looked from the top. This is just showing you how asymmetric this really is. You can see the DNA binds to one side only of this ring on the inside, and this subunit looks very, very different to this subunit. Let's look at this a little bit more. So you can see here a morph through all 6 different subunits going along the ring. This color scheme follows this, and you can see from this little dance here that the angle between the alpha and beta subunit, who themselves stay pretty much the same over that cycle, the angle changes by up to 34 degrees overall. So that's a very large conformational change for this class of proteins. And if you plot this angle, you can see it's sort of a wave function. So if you go around the ring, the beta subunit with respect to the alpha subunit, as is shown here, basically goes up and down, up and down, up and down. Okay. And as I showed you, the DNA sits on one side of the ring. So these 4 subunits are clearly binding to the backbone of the DNA. These 2 subunits are not. And what this means is, together, these 4 subunits that bind and their different angles between the alpha and beta subunits, it means that the beta subunits all bind to different positions along the helix of the DNA. So these 2 residues here bind to the backbone there, then 2 base pairs further, B binds to 2 base pairs further, A, and then F. So this is called the spiral staircase. This has been seen in other AAA proteins as well, but it is here especially pronounced and very, very clear. So let's just cut the chase and go to the mechanism. As I told you, we know the direction of how the hydrolysis works here. And we have the states of the 6 subunits. So the proposal here is now how this works is that it's not the subunits that rotates, but it's the states that rotate, right? So this subunit D would basically acquire the conformation of the subunit C in the next cycle, and that's shown here, right? So this is the yellow from that. So basically, all at each point of the 6 positions in the reaction cycle, each subunit will have one of the 6 conformation, and they all change at presumably the same time, although they don't. Doesn't have to be exactly the same time. So we can basically, from having this one structure and rotating the conformations around, we should get a movie of the entire process, how that works. And this is shown here, a little snippet of that. So this explains why we need to have this change in angle. So you can see here, 1, 2, 3, 4 attached. And then there's 2 subunits around the ring that don't attach for resetting. And then 1, 2, 3, 4. And then it detaches for two steps, and then it retaches again. That's why we need this change in conformation that goes around the ring. Okay. So in order to make this a bit clearer, we've made a fancy movie, spent some money on this to show you this in all its glory. Let's just hope this starts. So that's actually at very high speed. So we need to slow this down for you to be able to see anything. So this is a 1,000-fold reduced speed. This is 2,500-fold reduced from real-time speed, and this is 50,000 times reduced speed. And you can clearly see how the 6 subunits here change all their conformation at the same time. So 6 conformations rotating through the 6 subunits. ATP hydrolysis occurs in the blue subunit. This is now counter clockwise because we look from the other side. You can clearly see that the DNA now moves around the ring because it is only attached to 4 of the subunits at any point in time. The other 2 subunits are not in contact with the DNA. So this is showing again how these 6 conformations rotate around the ring. So it's basically the ring just made open, and you can see how each subunit goes through all these 6 states in a concerted reaction mechanism. And when that is finished, we can look at the DNA interfaces. We can see here that 2 reset, and then 1, 2, 3, 4 steps up, and then it detaches again and reattaches further down. And because each of these subunits holds onto 2 base pairs, we also know that the translocation step size is 2 base pairs. And this is sort of alternative explanation of this mechanism. You can also think of this as a treadmilling filament. Actually, you can see this red thing basically just treadmills around the DNA because there's always one edit at the leading edge, and there's always one removed at the trailing edge. So it's a great thing that the mechanism of this is related to what my group has been working on for many years on filament systems. It's basically a treadmilling filament that is closed into a ring. And we go back to the beginning. Right, that's all I wanted to tell you today. I hope that wasn't too long. I need to acknowledge a couple of people. So Francesca in my group has really been the star on the TG project that actually started off like so many things as a backup project, but I think it's a fantastic success for her. Tanmay has been the driver of the S-Layer project. He's now in Oxford taking that forward. And Nicola has really been the person who has worked most on the FTSK project, and Trevor has done some of the EM. I need to acknowledge some of our facilities here and everybody in my group, and thank you for your attention. And then before I stop, I just wanted to sort of go back to Max a little bit. We are very fond of him here, and as I said, we still have a restaurant like we used to have, and we have this little plaque in our lecture theater and his picture. And I just leave you with this most famous quote from him in science, truth always wins, and I hope, given our turbulent times, that that will still be true. And thank you for your attention.